They had been giving cocaine and methamphetamine to fruit flies and found that the same genes involved in circadian rhythm were involved in the behavioral response to these stimulants in the flies. So we were working with LSD in the lab. I thought, wouldn't it be really cool to give some LSD to flies and see what happens?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. My name is Jesse Lawler. I am your host, and this is the 207th episode of this podcast dedicated to anything and everything that you can do to help the better workings of your own brain, which, not surprisingly, oftentimes means getting down to the neuronal nitty-gritty and doing some hardcore basic science research to figure out why the brain works the way it does. And sometimes this means it's useful to grind up the brains in question, sometimes carefully dissect them, and sometimes just being able to count the number of neurons that are actually in there. But the one common element about all these research methods is that it pretty much means you're not going to be working on a living human brain. And that is where fruit flies often come in. And you can think of this episode as being sort of everything you always wanted to know about fruit fly brains but were afraid to ask. We're going to be talking about a great number of things, probably first and foremost serotonin and its role in the brain. And walking us through all these topics and about 20 years plus of his own research is Dr. Charles Nichols from Louisiana State University at New Orleans. We spoke with Dr. Nichols not long ago for the first time back in episode number 196, but that was sort of a cup runneth over interview. There was obviously more to be talked about, and we scheduled a follow-up, and that follow-up is what you're going to be hearing today. No less interesting than the first time around. If you hang around until the very end of the episode, keeping on these semi-psychedelic themes, I'm going to be talking about some new findings about the role of psychedelics and criminality, or perhaps better said as the prevention of criminality. We will see what the data says, but that'll be in the ruthless listener retention gimmick. As usual, though, let's first kick things off with This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
It's widely understood that people can be cognitively skilled at different levels in lots of different things. Some systems measure 7 different styles of intelligence, others more, others less. But one common theme in all of this is that regardless of the system, different types of intelligence tend to be correlated with one another. There probably is some oddball case in history where once somebody got 800 on the math SATs and 300 or whatever the minimum score is on the verbal SATs, but that would be an absolute anomaly, sort of the exception that makes the rule, because there's going to be a lot more people that when they do very well on one, they're going to do very well on the other. And the common underlying factor here, and in fact what IQ is supposed to give us a numerical measurement of, is something called general intelligence, or just G. If you wanted to know as much as you could about a person's general cognitive ability, but you could only know one piece of data, this would be the piece of data that you would want. How quickly you can learn to tie your shoes, your ability to write a sonnet or defuse a nuclear bomb, whatever it might be, general intelligence is going to be a factor. But researchers at Vanderbilt University have found an interesting exception to this general rule about general intelligence, and that is visual object recognition, which is definitely a cognitive process, interpreting medical x-rays or keeping track of various aircraft on a radar display, things like that. In a recent research paper entitled Domain-Specific and Domain-General Individual Differences in Visual Object Recognition, they showed 2 important findings. One is that there's a broad range of difference in the ability people have to perform these sorts of visual cognitive tasks. And the second thing is that these person-to-person differences seem to have no correlation with the general intelligence of the study participants. In other words, it would be no particular surprise to see that somebody's great at tracking aircraft on a radar display, but otherwise be dumb as a brick. To look at the variability in visual cognitive skills, the Vanderbilt researchers developed a new test which they call the Novel Object Memory Test, which allowed involving participants to study 6 target make-believe creatures. These kind of remind me of the little cartoon aliens in Toy Story that live in the machine that worship the claw that comes down and they say, "The claw." They're interesting computer-generated shapes, but they don't look like anything that exists in the real world. People are better at identifying novel car shapes if they have a good baseline familiarity with cars. Dog people have better recognition of dog breeds, things like that. So using these make-believe computer-generated monster sort of shapes, they put everybody on the same playing field. So in their basic test, each test participant would see 6 fake creatures that they're supposed to remember the shape of, followed by a number of trials where they would see a set of 3 similar creatures, one of which would be a match from the group that they were asked to memorize. The other 2 wouldn't match, they'd basically be decoys. And the test was for each group of these 3 make-believe creatures, could they pick out the one that should have been familiar to them? They ran this study on more than 2,000 subjects, and they found that there was a wide variation in people's ability. It was well conserved among people. So if you're good at memorizing instances of fake monster body style A, you would also be good at memorizing instances of fake monster body style B. And the actual variation in people's ability here, what you could think of as the width of the bell-shaped curve, was wider than the average spread of abilities on a standard nonverbal IQ test. Says Dr. Isabelle Gautier, one of the study heads. These findings are quite exciting because performance on cognitive skills is almost always associated with general intelligence. This suggests that we can really learn something new about people using these tests over and beyond all the abilities that we already know how to measure. A lot of jobs and hobbies depend on visual skills, and because they're independent of general intelligence, the next step is to explore how we can use these tests in real-world applications where performance could not be well predicted before.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
Picked up a couple of 5-star reviews on iTunes. Listener Elba Gista from the USA says, SDS has expanded my knowledge of nootropics and brain functionality and has allowed me to help countless people in my job and personal life. I'm officially the brain guy at my work now. And listener Troubled Speller, also from the USA, says, 5 big stars, and I wish I could give more. Well, thanks to both of you. Thanks to everybody that's been leaving iTunes reviews, sending links to friends, listening to the podcast far too loud in your open-top convertible. Every little bit counts, and I very much appreciate your seeding the podcast out there. If you have not yet signed up for the Brain Breakfast newsletter, you are cordially invited to do so. That is our weekly-ish newsletter, probably popping out a weekend edition this coming weekend. But if you want some oddball reading material, neuroscience news, and follow-ups and previews to the podcast episodes, you can get signed up for that at smartdrugsmarts.com/newsletter. It probably goes without saying right now that we're coming up on Black Friday and Cyber Monday and whatever they're calling the Saturday and Sunday in between. I don't know if those days have an official kind of name to go with them yet. Probably doesn't, I just don't know about it. But one thing that I do know is that there are holiday plans in store at both axonlabs.io and vitamonk.com. So if it's already Thanksgiving or beyond, definitely head on over there. There will be something holiday-ish happening. If you're hearing this early and you're hankering to buy something, but you don't want to lose out on the holiday deal, I will say without much fear of being contradicted that if you buy something a couple of days early and then some massive deal is released, they are not going to screw you out of that. There are good folks at both companies. Appeal to their holiday good cheer and you will not be disappointed. More breaking news on holiday supplement deals as such things become official, but do not hesitate to check out axonlabs.io and vitamonk.com for your nootropic and nutritional supplement needs for this holiday season and on into next year.
Smart Drug Smarts.
So I admit I've always felt a certain kinship, a certain positive bias towards fruit flies. They're flies, sure, but they don't seem to like dog poop and things like that. Instead, they'd kind of rather hang out in your kitchen eating fruit, which is something that I enjoy doing myself. And if you are even dimly aware of the huge body of scientific work that has been done on fruit flies and all the things that we've learned as a result, you'd probably get the idea that sharing some kitchen space with a couple of fruit flies every now and then is probably really a very small price to pay. To the species commonly called fruit flies, the scientific name is Drosophila melanogaster. They've got these beautiful ruby red eyes. We've got a really good close-up picture of one I was quite happy to find on the page for this podcast episode. But the scientist who I'm about to be speaking with, Dr. Charles Nichols, when we first spoke, he said his personal sort of version of Pascal's wager was whether there is in fact a fruit fly hell or a hell specifically reserved for the tormentors of fruit flies, because if there is, he's destined for it. We'll hope for his sake that's not the case. But he's been doing really interesting work studying pharmacology in the fruit fly brain and serotonin receptors in particular. Serotonin, a major neurotransmitter in humans, obviously, and in fruit flies also, it turns out. And many of the mechanisms by which serotonin is having its effects in the fruit fly brain do seem to relate to the way that serotonin works in higher animals' brains, including mammals and, yes, even humans. If you're a longtime Smart Drug Smarts listener, you know we've got our on-again, off-again series, Know Your Neurotransmitters. I was on the fence as to whether to actually make this a Know Your Neurotransmitters serotonin episode. It definitely deserves an asterisk and almost goes all the way there, but because we wind up talking about a lot of things, the brain's thalamus, overdosing elephants on LSD, lots of things that aren't directly relevant, I'm not quite willing to put the Know Your Neurotransmitters pin in this one, but you're going to learn a lot about serotonin by the time this interview is over. You were also going to hear the acronym CNS dropped a few times, just in case it's not obvious from context. CNS is central nervous system, so do not despair when acronyms come flying your way. I think CNS and LSD are the two acronyms you'll hear most in what comes next. So a very interesting interview coming up. Grab some overripe fruit or maybe some vinegar, and let's talk fruit flies with Dr. Charles Nichols.
The one thing when I tell people I work with fruit flies is they always ask, they say, oh, I've got fruit flies in my kitchen. How do I catch them? So then I end up telling him how to make a trap.
Okay, I'll bite. Just how does one catch fruit flies?
Well, fruit flies really like kind of fermenting fruit. So anything that has a little bit of yeasty smell or ethanol, alcohol to it. So if you have like an old beer bottle and leave about an inch or so of beer in the bottom of it so it gets all nice and stinky, and then you make a little funnel out of paper with just a opening at the end of it just large enough for a fly to get through, and then you tape that funnel into the opening of the beer bottle, and then you set those around, and so the fly will smell the yeast and the alcohol from the beer. Or you could use red wine. That works really well too. And then walk down the funnel, go through the little hole, and it'll be trapped in the bottle and can't get out.
Yeah, it sounds like one of those carnivorous plants, like the pitcher of death or something like that.
Oh, yeah. It's similar to that. But they work really well. And you can add a little bit of sugar to it to help revitalize the yeast so they're making more smells. You can add a little bit of vinegar. They like that smell too. But usually some old stale beer, red wine works great.
Okay, so an added bonus for all the fruit fly trapping enthusiasts out there. Back to the topic at hand. Bring us up to speed on the conceptual framework of using fruit flies to study the effects of psychedelics. And is there any translational potential between what we learn in a fruit fly brain, scaling that all the way up to something like a mammal or indeed a human?
Yes, I believe so. It was an interesting journey to get here. I think I mentioned previously that I had done my doctoral work in fruit fly eye and brain development, and then I never wanted to see another fruit fly again in my life.
You did say that, yeah.
I was just grinding up the flies. I was looking at their DNA on blots. It was really boring. So I wanted to do something mammalian. So I went into the psychedelic field at Vanderbilt in Elaine Sanders Bush's lab. And I was there for about 2 or 3 years, exclusively looking at the effects of LSD and methamphetamine as well on gene expression in rodent brains. And this paper came out from Jay Hirsch's group, who's at Virginia, and they had been giving cocaine and methamphetamine to fruit flies and did a genetic screen and found that the same genes involved in circadian rhythm, like clock, period, were involved in the behavioral response to these stimulants in the flies. And I thought that was really cool. The little light went off in my head, behavioral pharmacology in flies. It was like a new world opening up. So we were working with LSD in the lab, and there was a fly researcher who had a lab down the hall, Dr. B. Shea. So I thought, wouldn't it be really cool to give some LSD to flies and see what happens? Maybe we could find something interesting. And I went and talked to Elaine, and I pitched the idea of giving LSD to fruit flies to her. She thought about it for a minute and said, yeah, sure, go ahead. So I went down the hall, asked for some fruit flies, and the kind of fruit flies that I had wanted, the normal red-eyed flies, wild-type flies, they didn't have. So they gave me some white-eyed flies.
Just out of curiosity, why might one prefer a certain color of eye type of fly to another? Like, what's the difference there?
The usual wild type, the kind of fly that you'll see in the wild that has the most normal behavior, have red eyes. And the white mutant was one of the very first fruit fly mutants identified by Thomas Hunt Morgan back 100 years ago. And it's the one that is used as kind of the background to make all the transgenic flies in, because the transgenic gene elements are usually marked with the gene that gives the flies a red eye. So you start with a wide-eyed fly, and then you inject your DNA into the embryos, and after some breeding, you end up with red-eyed flies, and you know that your transgene is there because the fly now has red eyes.
Ah, interesting.
But other than the red eye color, they're fairly normal. With flies, you can't freeze them down, so you have to continuously put them in new food. And she had just done that to her red-eyed flies and didn't have a stock that had grown up yet for those. So I'm like, all right, I'll take the white-eyed flies. And how am I going to give them the drug? So I got some blotter paper and mixed up a sucrose LSD solution, put it on the blotter paper, starved the flies overnight so they were hungry and thirsty. And then the next day I put them on the blotter paper and watched them eat. They put the little proboscis down and sucked up food for about 2 or 3 minutes.
Yeah.
And like, okay, this cool is going to work. So then I just watched the fly to see what happened. I took him out of the little food chamber with the blotter paper, put him in another little, like a tiny petri dish so it wouldn't fly away. And after about 10 minutes, the fly stopped moving. I'm like, okay. So I took the top off the petri dish and I poked it, and it took a couple steps forward and didn't really move much more than that. And then a couple minutes later, he started to slowly fall over.
Never a good sign.
A slow motion fall over about 30 seconds onto his side. Like, oh, that's pretty cool. So I propped him back up with some little tweezers, and he fell over again, really slow. And so I propped him back again, and I poked him in the butt, and he took a couple steps forward and then fell over again. And then after about 20 minutes, he was just catatonic, non-responsive. And then after about an hour, woke back up and was fine. So I was like, oh, that's pretty cool. Flies have a response to LSD, so they must have serotonin receptors in them. And I had known that they had found serotonin in some receptors in the fly. This was in probably about 99 or 2000 that I was doing this. And I went into the literature and looked, and sure enough, flies had the target serotonin receptors for psychedelics, the serotonin 2 receptor, the serotonin 1A receptor. And then I dug a little bit deeper and found that the receptors had all been cloned. The DNA was known, but the man who cloned them had since left the fly world and had just characterized their expression in embryos. And no function at all was known of the serotonin receptors in the adult fly. And at that point, I was like, hey, this is a real opportunity here, is to develop the fly as a translational model for serotonin neuropharmacology. And to do that, we can use psychedelics and explore what are the effects of these drugs on the different behaviors. And so at that point in my postdoc, I started to make the transgenic fly tools to map where all the serotonin receptors were and start to develop some behavioral assays. And then, as it turned out, the next week, I went down and I got the red-eyed flies that I had wanted in the first place. And when I gave them LSD, there was absolutely no effect that I could tell.
Score one for serendipity.
Yes, definitely. The gene that is responsible for the red eyes in the fly instead of white is a transporter for the amino acid tryptophan. And so these flies that don't have the white gene, so their eyes are white, they're not getting tryptophan into the neurons. And if you're not getting tryptophan into the neurons, you're not making serotonin. And I think what's happening is what these flies had a serotonin deficiency, and they had an upregulation of the serotonin receptors and just happened to be super sensitive to psychedelics.
The question that I had was figuring out what the proper dosage of LSD for a fruit fly would be. I mean, that seems fraught with peril. Actually, I did a little bit of research before this interview on giving LSD to animals, and I came across this kind of heartbreaking story where I think this was in the 70s, they gave LSD to an elephant.
Oh, yeah, Jumbo.
You want to tell that story? I mean, that's sort of an interesting one from the history of animal LSD studies.
Oh, yeah. It was, if I can recall the details of this, it was back in the early 1960s. And you can get this paper out of Science magazine. It's on their archive, that they were studying a behavior called tusking in male elephants, which was some kind of male display courtship kind of behavior. And they thought that serotonin might be involved in it. So they had given this poor elephant a couple different drugs and then gave him this big giant shot of LSD. And then they describe in the paper that he's basically had a fit, went crazy, and keeled over in just a couple minutes after the LSD shot. And it had other drugs on board too. But it's funny reading that because it's in one of the top scientific journals. But, the scientific rigor of that experiment is just...
So the kind of thing that would never get approved today. I mean,
No. Trying to get that by your university's Animal Care and Use Committee, that just wouldn't fly.
Also, though, it's like they were trying to basically mimic psychosis in this thing where a male elephant goes into tusk. It's running rampant. They're kind of trying to figure out how to keep animals from doing this in zoos, I think was part of what their goal was.
Yeah, so they were trying to reproduce it with these massive doses of LSD, and they did something to it.
So if we know from that experiment historically that you would need a lesser amount of LSD to go into a larger animal per kilogram of body weight versus a human, does the opposite hold when you're scaling down to something like a fruit fly, that you would actually be using more LSD per kilogram of body weight? One
of the things with giving the drugs to the fruit flies is you can't do a blood draw and sort of see what levels they have.
Yeah.
So we have to rely on doing a dose response experiment where we go from a low dose that really has no effect to the maximal effect and seeing kind of what the half dose is that gave that effect. And that's usually or typically what we'll use in the experiments. And we have no idea what the actual concentrations in the fly are because we haven't had the equipment. You have to have some fairly high-end sophisticated measuring equipment to look at what these levels of drugs are in a fly brain.
But you do have a pretty good idea, I'm assuming, of how much water a thirsty fruit fly is going to drink.
Oh, yeah. When I first started dosing up the flies, I would spike the LSD solution on the blotter paper with a small amount of radioactive sugar. And then after the fly would do its behavioral assay, we would grind the fly up and then count it in a radioactive scintillation counter and then back calculate the volume and the amount of drug that the fly ate, sort of as a loose approximation. That proved to be really difficult, though, because we ended up having to take each of those flies and I would cut the wings off so they wouldn't fly away. They would just hop on the table and you could catch them much easier. And it was slow to do that. So when we give the drugs now, we do it primarily in the food and we keep the flies on the food with the drug for about 24 hours so they build up a steady state level.
So this was back in 1999-2000 when you were doing this first round of experiments.
Right. And the first thing we did was there was a lab in Germany that had found that some of the circuits in the fly brain that other researchers had shown express a high level of serotonin control the ability of a fly to follow moving objects in its environment. And what this lab in Germany has done, and a few other labs, is they have these really sophisticated computer-controlled flight simulators where you tether a fly in the middle of this environment and have it go to a computer, and it's surrounded by these basically 360-degree video screens. The fly is just kind of flying along in its virtual environment. You can throw objects in its path and see how it avoids it. But we did a really low-tech version of this, and we made a cylinder that has a stripe that moves around the outside of it. And a fly will normally follow a stripe, so it'll go in a circle and follow it. And we found that when we gave the red-eyed flies LSD, they were still just as active, but they couldn't follow the stripe. So it was interfering with their visual processing. That was our first paper on that. And we could rescue the effects of psychedelics with antagonists that block the receptor.
And the first fly you mentioned, you said came back to normal behaviorally after about an hour. That seems surprisingly fast. Are fruit flies metabolizing LSD quicker? Do they have less sticky serotonin receptors?
I think it's metabolic rate, but I did find that if I gave them enough LSD that they would not wake up. So there was a dose effect. But in general, humans in particular are much more sensitive to the CNS effects of psychedelics. With a rat, kind of a threshold dose, a good one used for drug discrimination is like 0.1 milligrams per kilogram, which in a human would be several milligrams instead of micrograms.
Wow, yeah.
So there's a scaling factor usually involved in like going to rodents. And humans, for a lot of drugs, are just more sensitive, and psychedelics are really more sensitive. And I think that's because of the unusually high density of the serotonin 2A receptors in the human brain compared to, say, a rat brain or a mouse brain, fly brain.
And if we were to look at a map of the human brain, would we find the density of serotonin 2A receptors more prominent in certain areas?
In the human or the fly?
The human.
In the human, in the prefrontal cortex, sort of the executive decision-making part of the brain is where it's the highest. But they're also at high levels in other nuclei, in the amygdala, paraventricular nucleus, where they regulate physiological autonomic function. They're in the hippocampus. There's a high density in the visual cortex. So a lot of the geometric patterns that people report after taking psychedelics are believed to be due to activation of the 2A receptors in the visual cortex at the back of the brain.
Yeah, I was wondering about that because such a huge proportion of the human brain by weight, by volume, is dedicated to visual processing. As compared to other animals, that's definitely sort of our primary sense. And I was wondering if that lopsidedness maybe had some relation to this extra serotonin 2A receptors.
That could be, because other animals like mice and rats, even dogs have a huge olfactory cortex, several hundred times better sense of smell than humans. And humans are primary visual. So the visual cortex, in and of itself, it's fairly large in the back of the brain, but it also recruits other areas of the brain for the processing of that and the integration into your conscious experience, rather than just sort of the pure reconstruction of the image, which happens in the back of the brain. But then your frontal cortex has to process that at the executive level and then decide what to do with that input. And a lot of that sensory information is mediated through the thalamus. That's kind of the gateway of the sensory information. Your auditory, your visual goes through the thalamus to the frontal cortex. And there's a high density of serotonin 2A receptors in the thalamus. And there's brain scans from Franz Wallenweiter have shown that the thalamus is activated in response to psychedelics. So the theory is that you are sort of removing the gate on the sensory input to the frontal cortex by activating the serotonin 2A receptors in the thalamus. And you're flooding the brain now with signals that normally would be filtered out. Little noises that would be in the background you wouldn't normally pay attention to suddenly are right there at the forefront with somebody you're having a conversation with.
That's interesting. That reminds me of the Aldous Huxley doors of perception concept of the brain sort of being a reality filter, or in this case, the thalamus maybe sort of being the morphological equivalent to that theory.
Yeah. And there's another brain nuclei that will assign meaning, like the locus coeruleus will assign meaning to sensory input. So now you've got enhanced meaning to sensory inputs that normally wouldn't have had a meaning. I think that all fits with what they're seeing now with the fMRI data, with the disintegration of the default mode network and nuclei talking to each other in the brain that don't normally talk to one another, that you're getting sort of this ungating of the thalamus, and it's just sending the inputs out to the cortex unfiltered. And then it's up to the cortex to really interpret that, and that's where a lot of, I think, the visual hallucinations will come in. It's your frontal cortex trying to make sense of all this information.
Yeah, and that really jibes intuitively with what it's like to be on psychedelics and just feel like the data streams have really been widened and you're kind of getting more than you can perceptually handle. It's really interesting, but also very disorienting.
I think with the therapy that they're doing for depression, anxiety at Johns Hopkins, NYU, and for cocaine addiction at UAB, it's this peak experience that they're having where they're connecting concepts outside the box that they normally wouldn't see on things in their life that may be contributing to where they currently are to put them in this depressed state or this addicted state. And once they see those connections outside the box, they're more able to effectively deal with them. And that psychedelics are really letting them see these connections outside the box in their life.
Sort of playing with this filtration idea, it would be really interesting if we were able to gain enough understanding of how the brain is doing these things to kind of keep the gross perceptual filters operating, but allow the conceptual filters to come off a little. So you would have a less confusing environment, but you could still kind of reframe things psychologically without some of the perceptual distortions.
Right. And that's kind of where the fruit fly comes in, because the fruit fly has the same neurotransmitters as humans. We've got the serotonin, we've got the dopamine D1, D2, serotonin 2A, 1A, acetylcholine, histamine. All the major neurotransmitters are there in the fly. And there are several different, if not hundreds of different circuits in the fly brain that are all communicating. And what we can do with the fly is give them the psychedelics and look to see how the different brain circuits are either activating or inactivating and how they are molecularly connected to and conveying their signal to the next circuit downstream. And we can begin to dissect how one circuit is communicating with another circuit when given a drug like, say, psilocybin. And then we can hypothesize that it's the same types of mechanisms that are occurring in, say, the human or the mammalian brain. But it would be a lot more difficult, if not impossible, to do a lot of those sort of really focused molecular types of experiments to look at the communication between brain circuits.
Now, would there be something equivalent to, say, the default mode network in a fruit fly? Or from what I understand on that, that's more kind of like the sense of self and identity and things like that you're trying to shut off when you meditate. Now, I wouldn't think that a fruit fly, whatever it has, it probably doesn't have a sense of self.
That's a very philosophical question. I don't know. I would say that the fly has a insect equivalent of a default mode network because you have the same types of excitatory and inhibitory systems. You've got the GABAergic systems that are inhibitory. You've got the glutamatergic, the cholinergic systems. You've got sensory inputs. You have auditory, you have olfactory, you have visual, you have memory centers, you have processing centers, circadian, and they all have to interact and communicate with one another. There are actually about 100,000 neurons in a fly brain that form all these structures. So if you could come up with an MRI machine small enough, you could probably find something akin to a default mode network.
Wow. There's a task for somebody listening out there, make a very, very small fMRI machine. So we sort of touched on a lot of the things, I guess, that have happened in the time since your graduate studies, but maybe walk us through a bit of also the areas where you might still be curious and you think that there's more to learn.
When I started, there was really not much interest in studying serotonin receptors. Because, at that time, most of the people in the fly world were doing a lot of genetic behaviors, developmental biology. Wasn't really much pharmacology going on, especially not with CNS active drugs. There were a few different groups There was a group studying alcohol at UCSF. At Virginia, they were studying CNS stimulants. I started studying psychedelics. And as I developed the tools, other groups were finding serotonin receptor by, say, looking at a behavior, say sleep, which involves serotonin, and they would do a genetic screen and say, oh, look, it's a serotonin receptor, and then come to it. So in the last 20 some years, there have been a lot of people who have come to the field of serotonin in the fly from whatever behavior or process they're looking at. And when I started, my first goal was to really find where these receptors were expressed in the fly. So I created transgenic flies that would express the green jellyfish protein under the promoter of the different serotonin receptor genes. And then using confocal microscopy, mapped out where the different serotonin receptors were in the fly brain. And then from that, we can guess maybe what sorts of behaviors they were involved in. And they're really all over the fly brain. The serotonin 2 receptor is in a small ring structure in the central complex that's involved in the fly equivalent of kind of cognitive processing. It's also involved in feeding and gustatory behavior. So I just started going through a lot of different behaviors to see what the serotonin receptors were involved in, using the psychedelics as a probe for that. So one of the first behaviors we looked at was aggression. If you take two flies and put them together, they'll fight. They can't really hurt each other, but they'll fence. They'll pounce on each other. They'll try to push each other off the food. About that time, there was a couple different groups who had demonstrated a role for serotonin in aggression, but they hadn't looked at the serotonin receptors. So we got the assay going up in the lab and got the flies fighting and started giving them different serotonin drugs, and saw that if we gave them a psychedelic that would activate the 5-HT2 receptor, that decreased the aggression in the fly. And if we gave them a more selective drug that just hit the serotonin 1 receptor, that they would get more aggressive. And so we thought, oh, psychedelics in flies at least are calming. We worked out a role for the different serotonin receptors in aggression. And then the next we looked at was learning and memory in the fly, that you can train a fly with Pavlovian conditioning to know that a shock has been paired with an odor. And it's got short-term memory, middle-term memory, long-term memory. And we looked at the different forms of memory with different serotonin drugs, psychedelics like LSD and psilocybin, dose response curves with that. And we established individual roles for these different receptors in the fly for learning and memory and how they're interacting. And then we moved on from that to look at courtship. And this specifically was with the serotonin 7 receptor. You put a male and a female fly together. Instead of fighting, they do this complicated dance through several stages that ultimately ends up in copulation. And we found that if we inactivate the serotonin 7 receptors, either genetically or with a drug, we prevented one fly from having interest in mating with the other fly. So it's kind of like the not tonight dear gene of the fly would be the serotonin 7 receptor. So we found that the different serotonin receptors are involved in really the fundamental processes of social interaction, with aggression, with courtship, with learning and memory. We did some work with circadian disruption. About the same time we were doing this, a group at UPenn had been studying the role of serotonin 1A in sleep. There's been a lot of study with that. They found a new serotonin receptor in the fly and published on that about a year or so ago, and that seems to be involved in feeding behaviors. So we know a tremendous amount more about what serotonin does in the fly now. So one of the things that I'm doing now is looking to see, now that we know the circuits and behaviors, how is that changing communication between other circuits, like between serotonin and dopamine circuits in the brain. So one of the main experiments that I'm doing now is trying to map the circuitry in the fly brain that responds to methamphetamine and cocaine, and then look at the role of serotonin and how serotonin is controlling how those different circuits interact with dopamine and other neurotransmitters. We can do that in the fly brain and really get at sort of the molecular mechanics of what's going on at the synapse between these different circuits really quickly and easily, instead of, if we could do some of these at all in a mouse, it would take months or years, and what we could do in just a few weeks.
Yeah, it seems like you've got E. coli and then like the worm C. elegans and then fruit flies and mice. And there's kind of like graduated steps up to full human studies. And yeah, fruit flies are definitely one of the major linchpins there.
The thing with fruit flies, unlike the worm like C. elegans, you can get really sophisticated behaviors. We can do with, say, CNS active drugs. We can do learning and memory, look at different aspects of that. We can do visual acuity. We can do conditioned place preference, self-administration, locomotor activity, grooming, courtship, aggression. We get these really complex, sophisticated behaviors that it's really the lowest organism that has defined behaviors that we can model that are relevant to what we see in humans.
As you were describing serotonin's effects on all these different fruit fly behaviors, I kept thinking, like, when we think of serotonin in humans, oftentimes one of the first things we think of is it has effects on mood.
Right.
Is mood even a concept that scales down to a fruit fly? Or should we think about this the other way, that mood is like sort of an epiphenomenon that we give a name to when we're talking about like a macro scale creature like a human, but really it's just kind of another way of mediating the behavior, which is really, what evolution thinks about?
That's a really good question, and something that I have a postdoc in the lab now who is trying to develop some behavioral assays to assess the mood and affect of a fly to see if we can do that. Because I'm curious to see if flies do have moods, if they can get depressed.
Yeah.
So we're actively looking at trying to develop some of these assays. And there are some people out there who have put flies in, say, an open field arena and looked at anxiety. An animal, so you put a mouse in an open room, it likes to go or in the corners in the side of the room. If you give it a benzodiazepine or something that is a treatment for anxiety, it gets more bold and will walk through the center of the room. And so there are a couple reports in flies that if you give like an SSRI, a drug like Prozac, to a fly, that it walks in the middle of an arena more so. One of the things that we can do with the fly is we can apply optogenetics or dread receptors to turn on or off neurons.
What are dread receptors?
Designer receptors exclusively activated by designer drugs. This is a technology developed by Brian Roth at UNC at Chapel Hill, where they're G protein coupled receptors, similar to the serotonin receptor, but they're acetylcholine receptors that he's mutated. So there's no activation from the endogenous neurotransmitter. The only thing that will activate them is this artificial drug that you feed to the animals that doesn't have effect on any other thing. So you can transgenically express a dread receptor in a particular neural circuit, feed the animal this drug, and either activate it or inactivate it. And it's on a slower time scale than optogenetics using lasers, but it's more useful for chronic treatment and conditionings. And so I've translated this technology to the flies and have been using it to activate and inactivate the brain circuitries. It's how we're looking at the mapping the circuitry that responds to cocaine and methamphetamine.
Reminds me of like the Manchurian Candidate for animals, something where you can flip a switch that nothing else is going to flip, except you've got the one remote control and no other biological process is going to mess with it.
Right. So we've been able to, with the dread flies, make them smell smells or not smell smells, increase or decrease activity, impair their memory, some neat stuff that we've been doing with it. There are some groups that have been using optogenetics in combination with these flight simulators and, turning on and off things and really remote control the fly. It's like there's a remote control brain implant for a cockroach that you could buy online and control a cockroach's movement with an app on your iPhone. You can make it go forward or backward or left or right. They sell it as a science kit experiment.
That is pretty amazing. A cockroach to call your own.
So they're doing those kinds of things, but more sophisticated with flies and genetics and lasers. So it is like kind of the Manchurian fly.
So where do you want to take this research next? What are you most curious about going forward?
I'm really curious to see if we can sort of understand the molecular control of circuits by serotonin receptors. Like in the mammalian brain, the serotonin receptors are modulating the function of the excitatory glutamate receptors, inhibitory GABA, that by activating serotonin receptors, they're either dialing up or dialing down the amount of other neurotransmitters like dopamine and glutamate that other neurotransmitter releasing neurons have, and how those are communicating between circuits. And so I really want to know, how does, say, giving a 5-HT2A receptor agonist influence how one brain circuit modulates, say, the dopamine release onto another circuit and how that responds? Because that will inform on, you're disintegrating the default mode network in humans. What does that really mean at the molecular level? And then when it reestablishes itself, how is that happening at the molecular level? We can't do that in humans. That will be nearly impossible to do in rodents, but it's something I think we could do in flies, is kind of get down at the interface between different circuits and get the molecular mechanisms of how psychedelics are doing that.
I think in the C. elegans, it's something like 300 neurons, and they've got basically sort of a working entirely virtual worm brain now. At 100,000 neurons in a fruit fly, how far are we from being able to kind of have a model fruit fly brain that we can mess with in silico?
Oh, I think we're a long way off. The C. elegans brain that they did was about 2 years ago. They built a physical model and then hooked it up without any programming to a Lego car with a sensor on it. And it would move forward and back and had some worm-like behaviors, and that was really cool. But that's only 300 neurons. And you scale that up to 100,000, and it just boggles the mind, even at that scale. I think we're a long way off. I think a fruit fly brain is probably more complex than a lot, if not all of the supercomputers that we have available out there in terms of what it can do. One issue I think is it's always been with fruit fly researchers is you go back to the comments of Sarah Palin several years ago. Why are we wasting federal research money on fruit flies? In my case, why are we spending federal money to watch gay flies mate? That was one of the experiments that we did for the courtship papers. One of the reviewers wanted to know if it affected male-male courtship.
Yeah.
And so we did that experiment, and the postdoc who did that experiment, that came up in her family. And so how can we, as scientists who are studying fruit flies, really convey the importance of basic science funding to experiments like this, and how looking at how fruit flies mate, how they respond to psychedelics, will really provide critical information into curing and understanding human diseases. And it's been an issue for decades. And I'm not sure if it's really making any progress or not, but the Nobel Prize in Physiology and Medicine this year went to 3 fruit fly researchers for their discovery, essentially, of the circadian clock, which was found first in flies and then in mammals. A lot of basic physiology, like the circadian clock, different circuitries, behavioral genetics, the development of transgenic animal technology was really developed first in the fruit fly. So, I always advocate for basic science of stuff like this because it really leads to the discoveries.
Smart Drug Smarts.
So a big thank you to Dr. Charles Nichols for taking the time for that conversation. I think we should all give a brief moment of silence for the many fruit flies that have unwittingly given their lives in the human pursuit of scientific discovery. They are all little fruit-eating martyrs. If you want to see a little animal boxing match between one fruit fly that's high on psychedelics and another that's doped up on antipsychotics, Dr. Nichols was kind enough to send a video over. We've got that up on the website, as well as some laser confocal microscope images of both fly eyes and fly brains. I found the discussion in there about the thalamus's role as somewhat of a switchboard of consciousness, a really interesting way of thinking about potentially what's happening under the effects of psychedelics to give the experiential effects that they do. But for right now, keeping things psychedelically themed, let's talk about pulling some patterns from the data in the ruthless listener retention gimmick.
Smart Drug Smarts, ruthless listener retention gimmick.
There are a few things nicer than having a scientific study that is actually highly powered enough. There's actually enough data behind it where you can be fairly confident in the conclusions. Typically, this means just having enough data points and enough consistency in the experimental results where you can be relatively sure statistical significance has been reached. And depending on what you're studying, you might be able to get a highly enough powered study with 50 people, 100 people, a couple hundred people. But every now and then you come across a study that has 500,000 data points, a truly remarkable number. And the findings are significant enough where instead of counting your lucky chickens that, well, we've got a lot of data here, there's almost a bit of a forehead slap of there was so much data available that's being gathered for so long. We probably could have looked at this back when we only had 100,000 data points and still found something pretty convincing and maybe done something with this information sooner. I can't help but sort of feel myself falling into the second camp when I think about a study recently published by researchers at the University of Alabama at Birmingham. The findings were published just this past September in the Journal of Psychopharmacology. The study was using data obtained by the National Survey on Drug Use and Health, administered by the U.S. Department of Health and Human Services, looking at data gathered over the past 13 years from 480,000, so just under 500,000 U.S. adult respondents, looking at the relationship between classical psychedelic use and criminal behavior. To fall under the classical psychedelic definition within this study, they were talking about ayahuasca, dimethyltryptamine, aka DMT, LSD, mescaline, peyote, San Pedro, which is a mescaline-containing cactus, and psilocybin mushrooms. Here's the interesting thing, and of course this is correlation, not causation, so don't draw undue conclusions, but among those some 500,000 people, people who had used a classic psychedelic were significantly less likely to have committed a crime or indulged in criminal behavior. Specifically, there was a 27% decrease in the likelihood that they'd committed a larceny or a theft, a 12% decrease in their odds of having committed assault, a 22% decrease in the overall odds of arrest for any property crime, and an 18% decrease in the odds of arrest for a violent crime within the past year. And the survey in question did not just ask about psychedelics, it asked about all sorts of drug use, and pretty much every drug that didn't fall under that classical psychedelics category, they saw the opposite relationship. Crime became more likely statistically if a person had indulged in a non-classic psychedelic drug. According to the study's author, Peter Hendricks, an associate professor in the Department of Health Behavior, these findings are consistent with a growing body of research suggesting that classic psychedelics confer enduring psychological and prosocial benefits. Given the costs of criminal behavior, the potential represented by this treatment paradigm is significant. Now, of course, one could also argue that there might be a hidden third factor. This could be more or less coincidence, and that people who are using psychedelics are too busy using psychedelics to commit crimes, or that non-criminals tend to enjoy psychedelics more. Dr. Hendricks, who I think it's fair to say probably hopes that the correlation is in fact a causation, points out that classic psychedelics can produce primary mystical experiences and have been used for millennia across cultures with therapeutic intentions.
Just say no to scratch that. Say yes to the Smart Drug Smarts podcast. Join our mailing list at www.smartdrugsmarts.com.
Okay, so that was it for episode number 207. Thank you for hanging around until the very end. If you enjoyed this episode, you can find the links to everything that we talked about here up online at smartdrugsmarts.com/207. And last week, if you missed it, we spoke with Dr. Draulio Araujo about the psychedelic beverage and potential medicinal compound that is still being studied called ayahuasca. And next week, we're going to be digging into the genes, talking about the study of personal genomics, the future of medicine, single nucleotide polymorphisms, what we currently know about how all these things affect the brain, and whether you should just get with the times and have a mandatory tongue swab policy before you agree to a first date. But I will see you back here next week, same time, same podcast, and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week in the meantime, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only, although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.